right? So under molecular basis of inheritance, already we discussed about the structure of DNA, right? About the nitrogenous bases and about the type of sugar and about the linkage that are present in the DNA and about the double helical structure of DNA. And also we saw about the packaging, because the length of the DNA is very large. It is 2.2 meter approximately, and it cannot fit into a small nucleus. So it has to be coiled or condensed to fit inside the nucleus. So that's what we discussed under the packaging of DNA. That is how the DNA is packaged into the nucleus, right? So today we will focus on the search for the genetic material. That is the experiments that were done to prove that the DNA was the genetic material, right? That is the transforming, what is the transforming principle? And the genetic material was DNA. And also we'll focus on the RNA world, that is what is the role of RNA, okay? So the first thing is we are going to discuss about the transforming principle. So what do you mean by transforming principle? So first we saw that Frederick Mischer, he discovered the nucleic acid, right? And he named it as nucleon. And then Mendel, he studied about the laws of inheritance. So all these were done in the earliest days. But none of them, they told that what is the transforming principle? So what is the reason for the transformation? So, or how what is transferred from parent to progeny? So further work by the other scientists, like we saw many other experiments by Margin and other scientists, they found that is, they studied a lot about the chromosomes and they narrowed down the research to the molecular level, but the DNA was the genetic material was not identified. So the first experiment to prove the transforming principle was by Griffith. So Griffith's transforming principle experiment. So this was the first experiment in 1928 to prove that something was transferred, right, from one organism to the other organism. So what he did is, Frederick Griffith in 1928, he used mice and the Streptococcus pneumoniae. So he found that the Streptococcus pneumoniae has two strains. One is called the S strain, the other one is called the R strain. So what is the difference between S and the R strain? So this S strain, which is also called the smooth strain or the virulent strain, right? So virulent means it can cause disease. So this has a polysaccharide mucus coat and it causes pneumonia. So this Streptococcus pneumoniae is the organism which is responsible for causing pneumonia. So this has a polysaccharide mucus coat. Then there exists another strain, that is the R strain, which is non-virulent. So here there is no mucus coat and it did not cause pneumonia. So this is the A strain which has a coat surrounding the organism, right? So this is the A strain or the smooth strain, and this is the R strain or the rough strain. So this is the virulent strain and this is the non-virulent. So this is the virulent strain, okay? So what he did is, when he injected the S strain into the mice dies because the S strain is the virulent strain and it is capable of causing the disease. So the mice die. And when the R strain is injected into the mice lives because it is not virulent and it is not causing the disease. So the mice live. Then A strain, which is heat killed, he used the heat killed A strain and it is injected into the mice lives. So when A strain is injected as it is, the mice die. When it is injected with the heat killed A strain, the mice lives. So what is happening here? When the heat kill, that is when it is exposed to the high temperature, it deactivates. So the disease causing capacity, right? So when it is injected into the mice lives. But when he used the A strain, which is heat killed, with the R strain, that is live R strain, and it is injected into the mice, what happens? The mice dies. So what is the difference between the two? So here, even though it is heat killed, some factor is transferred from A strain to the R strain, right? Some factor, but he was not sure what is transferred because the biochemical nature of the factor that was transferred was not identified. But he came to one conclusion that some factor was transferred from A strain to the R strain. So when it is injected into the mice dies. Okay, so this was the Griffith transforming principle experiment. Okay, so rough strain, which is a non-virulent strain, when it is injected, the mouse lives, and smooth strain, which is a virulent strain, so the mouse dies, and heat kills smooth strain, so that is the structure is altered, right? The mouse lives, and the rough strain and the heat killed smooth strain. So here the mouse dies. Okay, so this is the Griffith's transforming principle experiment. So the conclusion, so what is the conclusion? He concluded that some transforming principle, that is some factor, is transferred from heat killed S strain to R strain. So something is transferred from S strain to R strain. And this enabled the R strain to synthesize the smooth polysaccharide coat. So it is having a smooth polysaccharide coat outside, right? So this transformation enabled the R strain to synthesize the smooth polysaccharide coat and become virulent. So it is responsible for the virulent nature. So it is due to the transfer of genetic material. So this is called the transforming principle experiment, but Griffith was not sure what was the transforming principle, whether it is a DNA or RNA or a protein. So something is transferred. Okay, so you're clear with this experiment. So then further work by the other scientists, they did the biochemical characterization of transforming principles. So what is responsible for this transformation? That is, when a heat kill A strain, it's injected with the live R strain. That is, there occurs in some transformation, right? And it caused the mouse to die. So what was the transforming principle? So that was further studied by the scientists, that is Oswald Avery, Colin MacLeod, and Macklin McCarthy. So these three scientists, they worked to determine the biochemical nature of the transforming principle in Griffith's experiment. Okay, so they worked on the Griffith's experiment. So what they did, they purified the biochemicals. That is, they separated the proteins, DNA, and RNA from heat-killed S cells using suitable enzymes. So what they did, so these are the heat-killed S cells. So they add different enzymes like proteinase, RNase, and DNase. So what is the role of proteinase? It will cleave the protein. RNase, that is ribonuclease, it will cleave the RNA. DNase, deoxyribonuclease, will cleave the DNA. So what they did, they purified these biochemicals, right, using suitable enzymes. So here it will affect only the protein. So here then they added the R cells. Okay, to this they added the R cells, and S cells appear in this experiment. The S cells again appear in the flask which is treated with RNAs. Here also there occurs an appearance of S cells. But in the case of the flask with DNAs, here there was no appearance of S cells. There is no S cells appear. So what is the basic principle behind this? So they discovered that digestion of proteins and RNA using proteases and RNases did not affect transformation. So the transformation was not affected in those test tubes. The addition of proteins and RNAs, because they affect only the protein and the RNA. So the transforming substance was not a protein or an RNA, but in the tube which was treated with DNAs, that inhibited the transformation. Okay, so this means that DNA caused transformation of R cells to S cells, and DNA was the transforming principle. So first in Griffiths, so you have to be clear in what experiment, who identified what is the principle, or what is the thing that is identified by the particular scientist. So Griffith, he found only the transforming principle, but he was not sure whether it was a DNA, RNA, or a protein. But further experiments by Avery McLeod, then McCarthy, they found out that the transforming principle was DNA. So how they found it? By using the different enzymes like proteases, RNases, and DNases. So when it is treated with proteases and RNases, so if you see, there occurs an appearance of the cells. That is, the cell growth was not affected. But here, since the DNA itself is affected, there was no transformation and there was no S cells, because that DNA was not transferred here. Okay, because it was treated with DNase enzyme. So by this experiment, they found that the transforming principle was DNA. Okay. So you are clear, right, till now, in these two experiments about Griffith and how it was found, that is biochemical characterization by Avery, McLeod, and McCarthy. Okay, clear? So Nandini and Himanshu, clear, right? So we'll go to the next experiment. So then the next experiment was by Hershey and Chase. So what is Hershey-Chase experiment? So this is also called blender experiment. So what they did is, Hershey and Chase, they grew some bacteriophage viruses. So what are bacteriophages? So bacteriophages are nothing but the viruses that affect bacteria. So what they did, they grew some bacteriophages on a medium containing radioactive phosphorus. So what is the purpose of adding radioactive phosphorus? So that wherever phosphorus is present, we can easily detect by means of radioactivity. So they added the radioactive phosphorus, there is P32, and some others on a medium containing radioactive sulfur. So this is the bacteriophage that contain the P32 DNA, and then here this is a T2 phage or grown in a medium containing S35, that is sulfur which is present in protein, but the phosphorus will be present in DNA. Okay, so what they did, first they grew some bacteriophages in a medium containing P32 and some other on a medium containing S35 in two separate devices, that is in flasks. Okay, then the viruses which are grown in P32 got radioactive DNA. So here these viruses, they have radioactive DNA because the phosphorus is incorporated into the DNA. You know the structure of DNA, right? There occurs the nitrogenous base and then sugar and phosphate. So this phosphorus is now radioactively labeled. So the viruses grown in P32 got radioactive DNA because only DNA contains phosphorus. And viruses which are grown in S35, they got radioactive protein because sulfur is a part of a protein, right? So the viruses grown in S35, they got radioactive protein. So now what they did, actually they grew this viruses, and then the label viruses are used to infect bacteria. Now this grown viruses are used to infect bacteria. So what is the next step? So these preparations, so what they prepared here, right? So this is the preparation, that is they made it to grow, made the virus to grow with the P32, and they also made the virus to grow with S32. So the next step is these preparations were used separately to infect E. coli. So they used this to infect the E. coli. So now after infection, the E. coli cells were gently agitated in a blender. So what is the purpose of using blender? For example, if you see the experiment, so now this is the bacteriophage. Here it is radioactive protein capsule. So the outer part is there, right? This red one, this is the capsule. So this will be labeled with S32, sorry, S35. And here the another set is, this is radioactive P32 labeled in it. So now inside the red, right? So this is P32. So they are doing in a two separate experiments, right? So here now they are making this bacteriophage to infect the E. coli. So this is the E. coli. So these bacteriophages, now they infect the E. coli. So that is the first step. First is the preparation. After preparation, they are made to infect the E. coli. So infection, then blending. So what is the use of this blending? So after the, what happened, this nucleic acid, it enters into the E. coli, right? It enters into the E. coli, only the nucleic acid enters. So outside, the protein coat will stay outside. So this are all protein coat, protein capsule. So blending, what is the role of the blending now? To remove the virus particles from bacteria. So whatever the things which are lying outside, so all these will be, these are the virus particles. So by blending, these virus particles will be removed. Okay. So then the culture was centrifuged to separate the lighter virus particles from heavier bacterial cells. So the bacterial cells are heavy, but the virus particles, that is the outer, the protein coat, everything will be, first we have to separate it. For that they use the technique centrifugation. So what is the use of centrifugation? It will separate the particles based on the molecular weight. So whatever is the lighter particle will be separated, and it will be separated in the supernatant or the liquid that is present above, right? So they centrifuge to separate the lighter virus particle from heavier bacterial cells. Now the bacteria infected with viruses having radioactive DNA were radioactive. That is, the DNA has passed from virus to bacteria. So if you observe it, so this is the blending. So all these will be separated. Now only the bacterial cell exists. So after centrifugation, no radioactivity is detected in cells. So if you observe this set of experiment which is treated with S35 labeled. So here if you check inside the cells, no radioactivity was observed because the S35 will be present only in the protein, right? So these proteins are present only outside and it is not entering into the cell. So, but the radioactive S35 was detected in the supernatant. So supernatant in the sense, if you are centrifuging, what will happen? The upper supernatant liquid will have the lighter particles, so that contain the radioactive sulfur, because all the proteins, these virus particles will be present in the upper layer. But if you check in the next set of experiments, that is radioactive P32 detected in cells. So here the radioactivity was detected because the DNA enters into the cell. Okay. But no radioactivity is detected in the supernatant. In the liquid, there was no radioactivity because only the protein coat is outside. So this phosphorus, they entered only into the DNA and not in the proteins. So no radioactivity was detected in the supernatant. Okay. So bacteria infected with viruses having radioactive DNA are radioactive. So here the bacterial cell itself is radioactive because the DNA has passed from virus to bacteria. So it entered from virus, it entered into the bacterial cell. Okay. But the bacteria which is infected with viruses having radioactive proteins were not radioactive. So these bacteria, they were not radioactive because the proteins were present only outside and they did not enter into the bacteria. Okay. So are you clear with this? This is called the blender experiment. So it involves 3 stages: infection, blending, and then centrifugation. So clear, right? So the next is the properties of genetic material. So if it has to be a genetic material, so what are the properties that a particular genetic material should have? That is whether it is a DNA or a RNA. So the genetic material must have the following properties. It should have the ability to generate its replica. That is, if the genetic material is transferring, that is, if it is transferred from parent to progeny, here they must be able to generate its replica, or it should be capable of undergoing replication. So that is one of the important property, right? And it should be chemically and structurally stable. So only if it is stable, it will be, that is, it can pass its information from parent to the offspring, right? So it should be chemically stable and structurally stable. And it provide the mutations which are needed for evolution. The mutations must be slow such that there occurs an evolution. And it should be able to express as Mendelian characters. So it leads to variation. So these are the general properties of a genetic material. Okay. So why the DNA is a stable genetic material? It's because we know that DNA is the genetic material in most of the organisms, right? So RNA is a genetic material only in some viruses, but DNA only is the genetic material in most organisms. So what are the reasons for stability of DNA, or why it is less reactive? And what are the reasons for mutability of RNA? That is, it is more prone to mutation. The RNA is more prone to mutation. So what is the reason for the high mutability or the high reactivity for RNA? So the main reason for stability here is it is double-stranded. We know that the structure of DNA is a double helical structure. So the reason for the stability is one is double-stranded, right? And the second reason is due to the presence of thymine. It has the thymine as a nitrogenous base instead of uracil. And also absence of 2-hydroxyl in the sugar, because this has a deoxyribose sugar which doesn't have the hydroxyl group at the second position. And that is also one of the reasons for less reactivity. But if you take the RNA, it is single-stranded, so it is not much stable. But here it follows the Chargaff rule, and the stability is more. There occurs an hydrogen bonding, and the stability is more when compared to that of RNA. And here it shows the presence of uracil instead of thymine, and also presence of 2-hydroxyl group in the ribose sugar. So that is also one of the reasons for the high reactivity. That is, its reactivity rate is high. So DNA is more stable when compared to RNA. So RNA is unstable, right? We know that there are many viruses which contain the RNA as a genetic material. For example, even if you take coronavirus, what is the genetic material there? It is only the RNA. So actually there are several mutant form exist, right? So in the case, if it is in the form of a DNA, the mutation rate will be less because it is stable. So RNA is the genetic material and it evolves at such a fast rate. So the mutating ability is very high. So even we are hearing every day that mutant strains are arising, right? So example for an RNA virus is a coronavirus even. So, and the other RNA viruses are QB, bacteriophage, and then tobacco mosaic virus, they mutate and evolve faster. So the mutation rate is very high and it is so unstable. The DNA strands are complementary. So if you take the stability of the DNA here, you can find the complementary strands, right? So on heating, they separate. So when the DNA strands are exposed to high temperature, they separate. But in appropriate conditions, they come together. So even in the case of Griffith experiment, what we saw, that is heat killed S strains, right? So when it is injected into the mouse, what happens first? The disease causing ability is lost. But when it is treated with the R, that is live strains, so there occurs some transformation. So what is that due to? Even when it is heat killed, at that time the disease causing ability is lost, but it can regain. For example, when the DNA is heated to high temperature, what happens? There occurs a denaturation, but when appropriate conditions resume, they come together, they can be converted to a double helical structure. So first by denaturation, this double helical structure will be converted to a single stranded structure, but under suitable conditions, again this single stranded structure can be converted into a double helical structure. That's what he observed in Griffith experiment. That is, some properties of DNA of the heat killed bacteria, they did not destroy. That is, even after heating with at high temperature, some of the properties were not destroyed, right? So this indicates the stability of the DNA. So DNA is stable when compared to that of RNA. Okay. So when you consider the characteristic of that is storing of genetic information, that is, what do you mean by storing of genetic information? That is, data is transferred from parent to progeny and it is transferred for further generations, right? So for the storage of genetic information, DNA is preferred due to its stability. But for the transmission of genetic information, RNA is better. So what do you mean by this? But what is the role of RNA in a cell? DNA, you're saying that it is the genetic material. So what is the role of RNA in the cell? So in some viruses, it acts as a genetic material, but generally in other organisms, it acts as a messenger, right? That is the messenger RNA. It helps as a messenger, that is, it carries the information for the synthesis of protein, right? So messenger RNA and then tRNA. What is the role of tRNA? It acts as an adapter molecule. It helps in the transport of amino acids for the synthesis of proteins. And then another RNA is rRNA. So this is a constituent of the ribosomes and it is present in large number when compared to that of mRNA and tRNA. So for the transmission of genetic information, RNA is better. So how the genetic information is transferred? So normally this DNA, so what is the central dogma actually? Central dogma is first the DNA, which is converted to RNA and this is converted to protein, right? So this DNA is replicated, that is DNA to DNA is replication. And the DNA to RNA, the process is called transcription. And RNA to protein, the process is called translation. And in some viruses, what happens? This RNA is converted to DNA. So it is called reverse transcription. That is the reverse of the transcription process. It's called reverse transcription. Okay. So this RNA, it can directly code for protein synthesis. So this RNA can be directly used for protein synthesis and can easily express the characters. but DNA is dependent on RNA for protein synthesis. So first the DNA has to be converted to RNA and then it has to be converted to protein. Okay. So as far as the storage of genetic information is concerned, DNA is preferred. So for the transmission of genetic information, RNA is preferred. That is, it acts as a messenger. Okay. So any doubts in this till now? Or we'll discuss few questions. Clear? Okay. So what do you mean by RNA world? Actually, RNA was the first genetic material. So it acts as, and also it acts as a catalyst. That is RNA, it also acts as a catalyst in some cases. Okay, and also if you take the essential life processes like metabolism, translation, and splicing, which all these processes, they evolved around RNA. So RNA is mainly involved in the translation, that is to form proteins, right? So DNA evolved from RNA. So they came to a conclusion that the DNA evolved from RNA for stability because DNA is more stable when compared to that of RNA. Okay, so RNA was the first genetic material that was formed. So what is the finding of Rizee and Chase? The viral infecting agent is RNA, the viral infecting agent is DNA, the viral infecting agent is protein. The T2 virus has RNA as its genetic material. So what was the finding of Rizee and Chase in their experiment? Yes, B is the right answer. That is Rizee and Chase, they found that the viral infecting agent is DNA. So B is the right answer. The transforming substance of pneumococcus in Griffith's experiment was protein, RNA, DNA, and then polysaccharide. Yes, again DNA. DNA is the transforming principle there. In 1928, Frederick Griffith showed that the hereditary material can pass from one type of bacteria into another through a process called transduction, invasion, recombination, transformation. Yes, transformation is the right answer. The structures in chromatin seen as bead on string when viewed under electron microscope are called nucleotides, nucleosides, histone octamers, nucleosomes. Yeah, nucleosomes is the right answer. That is structures in chromatin seen as bead on string. So that will look like a string with beads on it. So here these structures are called nucleosomes, right? They are the histone octamer which are wrapped with DNA. So nucleosomes is the right answer. Euchromatin is loosely packed, stains light, transcriptionally active, early replicating. So which is the right answer? So which are all the correct options for euchromatin? A and C, A, B, C, D, B, C, D, A, B, C. Yes, B is the right answer. So all are correct options. That is, euchromatin is loosely packed. Euchromatin stains light and it is transcriptionally active form of chromatin. It is early replicating, so it replicates in the early S phase, but heterochromatin replicates later in the S phase. So this is also a right statement. So all are correct statements, so B is the right option. Nucleic acids are made up of nitrogenous bases, amino acid, sugar, and phosphate. So which is the right option here? Nucleic acids are made up of, yes, A is the right answer. That is, nucleic acids are made up of nitrogenous bases, sugar, and phosphate, and not amino acids. So A, C, D, A is the right option. Hershey-Chase experiment determines that protein and DNA are the hereditary materials of viruses. Protein, not DNA, is the hereditary material of viruses. Viruses do not contain hereditary material. DNA, not protein, is the hereditary material of viruses. Case D is the right option. So I hope you're clear with these topics, right, about the structure of DNA and about the search for the genetic material. So what about Himanshu? Are you clear? Yes, so we'll discuss a few more questions. And actually, I sent you questions for principles of inheritance and variation. And did you try it? Okay. So did you try those questions? I posted it today. Yeah, in Botany. I posted it in the Botany group. Yeah, you check it and you try. So you didn't see, Nandini? Yeah, I posted it in the morning. So you check those questions. And if you have any queries, maybe we'll discuss answers tomorrow. Okay. Okay, you complete those questions. Maybe we'll discuss the answers tomorrow if you have any doubts, and maybe we can check the answers tomorrow. Okay, so now we'll discuss some more questions related to the structure of DNA and these experiments. The first question, what happened when heat kill S cells along with live R cells were injected into the mice? Option E, mice survived and showed live S cells, and option B, mice died and showed live S cells, and option C, mice survived and showed live R cells, and option D, mice died and showed live R cells. Is B is the right answer. That is, mice died and it showed the presence of live S cells. B is the right answer. Then the result of which of the following reaction experimentally carried out by Avery et al. On Streptococcus pneumoniae has proved conclusively that DNA is the genetic material? First, option A, live R strain plus DNA from S strain plus RNAs. And option B, live R strain DNA from S strain plus DNAs. And option C, live R strain plus denatured DNA of S strain plus protease. And option D, heat killed R strain plus DNA from S strain plus DNAs. So, which is the right option? C. Oh, it's not C. Actually, got confused, I think. It is actually option B, which shows that is it as live R strain plus DNA from A strain plus DNAs. Actually, this is the right option, because live R strain plus DNA from A strain, when it is treated with DNA's enzyme, it shows that DNA is the genetic material. But in the case of option C, it is given as live R strain plus denatured DNA of A strain plus protease, it is given. Okay, so actually this is not the right option. So option B, this is the right option when it is treated with DNAs. Okay, so I hope you are clear now. So B is the right answer when it is treated with the DNase enzyme. Clear? We'll go to next question. Then in Hershey-Chase experiment, radioactivity P32 was used to culture bacteriophages, which resulted in radioactive. Option A, viral DNA. Option B, bacterial capsule. Option C, viral proteins. Option D, plasma membrane of bacteria. Yes, A is the right answer because the P32 label will be present only in viral DNA. Okay, so A is the right answer. A molecule can act as a genetic material, must fulfill the traits given below, except option A, it should be able to express itself in the form of Mendelian characters. It should be able to generate its replica. It should be unstable structurally and chemically. It should provide the scope for slow changes that are required for evolution. Yes, option C is the right answer because it is given that it should be unstable, but the genetic material should be stable. So C is the right option. Then DNA is more stable than RNA because option E, DNA is double-stranded. Option B, thymine is present. Option C, 2'-hydroxyl is absent in deoxyribose. D, all of these. Yes, D is the right answer here. Then select the odd one. Adenosine, guanosine, cytosine, and thymidine. So which is the odd one? Adenosine, guanosine, cytosine, and thymidine. So adenosine, guanosine, cytosine, and thymidine. So which is the odd one here? No, it's not D. The odd one here is cytosine. Why it is odd one? Here adenosine is a nucleoside. This is also a nucleoside, but cytosine is a nitrogenous base. Okay, so all these are nucleosides, but this is a nitrogenous base. So cytosine is the odd one out here. So you have to be clear with these terms of nucleosides, nucleotides, and nitrogenous bases. Okay. Then the correct combination that can form an RNA nucleotide is option A, adenine plus deoxyribose plus phosphate. Option B, thymine plus ribose plus phosphate. Option C, uracil plus deoxyribose plus phosphate. Option D, uracil plus ribose plus phosphate. So which combination it can form an RNA nucleotide? Is option D is the right answer because in option A it is given deoxyribose. So deoxyribose will not be present in RNA. In option B it is given thymine. Thymine will not be present in RNA. And in option C it is given uracil plus deoxyribose. So deoxyribose is not present in RNA. So option D is the right answer, which has uracil, which is present only in RNA, and ribose sugar, which is present only in RNA, plus phosphate. So this is the correct combination of RNA nucleotides. So you have to be clear with this. In some viruses, RNA is present instead of DNA, indicating that option E, the nucleic acid must combine with the host DNA before replication. They cannot replicate. There is no hereditary information. RNA can act to transfer heredity. So what is the right option here? The RNA is present instead of DNA, indicating that option D is the right answer. That is, the RNA can act to transfer heredity. So that's why it is present in some cases. So it can act to transfer hereditary in some viruses. Okay. And the next question, DNA double helix model is proposed based on X-ray diffraction data produced by option A, Frederick Mescher, option B, Watson and Greek, then option C, Wilkins and Franklin, option D, Erwin Chargaff. DNA double helix model is proposed based on X-ray diffraction data produced by option A, Frederick Mixture. Option B, Watson and Crick. Option C, Wilkins and Franklin. Option D, Edwin Chargaff. Yes, option C is the right answer. That is Wilkins and Franklin. And Franklin. So they propose the X-ray diffraction data. Diffraction. Okay. So you have to be clear with these scientists. Frederick Mixture, that is nucleic acid, and then Watson and Crick double helical model. So X-ray diffraction by Wilkins and Franklin. So some of the common scientist name, you have to be clear with it. Okay. Then DNA of E. coli has option A, it is 4.6 into 10 power 6 base pairs. Option B, it is 6.6 into 10 power 9 base pairs. And then option C, it is 5,386 nucleotides. And option D, it is 48,502 base pairs. So which is the right option? Is option A. Option A is the right answer. That is DNA of E. coli has 4.6 into 10 power 6 base pairs. Then nucleotide arrangement in DNA can be seen by X-ray crystallography, electron microscope, ultracentrifuge, and light microscope. Yes, actually the correct answer here is X-ray crystallography. X-ray crystallography. That is the nucleotide arrangement can be studied by X-ray crystallography. A is the right answer. Okay. Then the next question is, a short length of a DNA molecule contains 120 adenine. A short length of a DNA molecule contains 120 adenine and 120 cytosine. So the total number of nucleotides in this DNA fragment is 120, 60, 240, and 480. So which is the correct answer? The short length of a DNA molecule has 120 adenine and 120 cytosine. So the total number of nucleotide is D is the right answer. So how will you find out this? So 120 adenine means, that is adenine is 120. So thymine will also be 120. If cytosine is 120, then guanine will also be 120. So there occurs a bonding between guanine and cytosine and adenine and thymine. So totally there will be 480. So 480 is the right answer. You're clear, right? So the total number of nucleotides in this DNA fragment is 480. Then next, which is correct statement? DNA and histone are positively charged. DNA and histone are negatively charged. And option C, DNA is negatively charged and histone is positively charged. And option D, DNA is positively charged and histone is negatively charged. Yes, C is the right answer. That is, DNA is negatively charged due to the presence of phosphate and histone is positively charged due to the presence of basic amino acids. It is lysine and arginine, right? So C is the right answer. Then the negatively charged DNA is wrapped around histone octamer to give nucleotide, euchromatin, nucleosome, heterochromatin. Yes, it is nucleosome. Nucleosome is the right answer. That is the DNA with histone octamer. It is called a nucleosome. The association of H1 histone with the nucleosome indicates that option A, DNA replication is occurring. Option B, DNA is condensed into a chromatin fiber. Option C, the DNA double helix is exposed. Option D, transcription is occurring. So which is the right answer here? Yes, B. The association of, that is histone H1. Association of histone H1 with nucleosome. So what does this show? It shows that the DNA is condensed into a chromatin fiber. Is condensed to form a chromatin fiber. Okay, so this is the right answer. And in sea urchin DNA, which is double-stranded, so that is that sea urchin DNA is double-stranded. 17% of the bases was shown to be cytosine. The percentage of other bases expected to be present in the DNA are option A, G34, A 24.5% and T 24.5%. Then option B, G17, then A 16.5, T is 32.5%. And option C, it is G17, A 33, T 33. And option D, it is G, which is 8.5%, A 50%, and T 24.5%. So which is the right option? Yes, C is the right option, because when C is 17%, obviously G is equal to C, right? So G is also 17%. So what is the value of G plus C? That is 17 plus 70. So what is the value? Is 34, right? So A plus T will be 66%. So among this, so A will be 33% and T will be 33%. Clear? So you have to be clear to calculate these type of problems. Okay. So C is the right answer. That is G is equal to 17% and A is equal to 33% and T is equal to 33%. So clear? So are you able to follow Himanshu? You can give your answers in the chat. So the next question, which one is not applicable to RNA? Complementary base pairing, 5' and 3' ends heterocyclic nitrogenous bases and Chargaff rule. So which one is not applicable to RNA? Yes, D. Chargaff rule is applicable only to DNA, where the adenine content will be equal to thymine. So these rules are applicable only to DNA and not to RNA. Then transformation was discovered by option A, Michelson and Stahl, option B, Hersey and Chase, option C, Griffith, option D, Watson and Greek. Transformation was discovered by, so first transforming principle was discovered by Ace, it is Griffith. So you see the term clearly, the transforming principle was explained or discovered by Griffith. Then Hershey and Chase. Then next is Avery, MacLeod and McCarthy. They found the DNA as the genetic material. Then Hershey and Chase, they found that the DNA which enters into the cell and it is responsible for the infection and not the, that is the DNA by using the radioactive phosphorus and sulfur. So sulfur is present only in the protein and this phosphorus is present inside the DNA. So DNA is what it enters into the cells, right? So that was proved by Hershey and Chase. So the next question is, so how far is each base pair from the next one in DNA double helix model? Option A, it is 2 nanometer, option B, it is 3.4 nanometer, and option C, it is 34 nanometer and 0.34 nanometer. The question is the distance between each base pair. The distance between each base pair. Yes. It is 0.34. So be clear with the question. Okay, then you answer. D is the right option. It's each base pair from the next one. Then find out the wrong statement about heterochromatin. It is densely packed, it stains dark, it is transcriptionally active, it is late replicating. Find the wrong statement about heterochromatin. Yes, it is transcriptionally active is a wrong statement because only euchromatin is transcriptionally active form of the chromatin. So C is the right answer. The year 2003 was celebrated as the 50th anniversary of discovery of option A, transpositions by Barbara Clintock. Option B, structure of DNA by Watson and Crick. Option C, Mendel's law of inheritance. Then option D, biotechnology by Harry Miller. So the year 2003 was celebrated as the 50th anniversary of discovery of S. Option B is the right answer. That is, it is the structure of DNA by Watson and Crick. Then a typical nucleosome contains 100 base pairs of DNA helix or 400 base pairs of DNA helix or 200 base pairs of DNA helix or 300 base pairs. Yes, again option C is the right answer because the nucleosome contain 200 base pairs of DNA helix. Then, which of the following are the functions of RNA? What is the function of RNA? Option E, it is a carrier of genetic information from DNA to ribosomes, synthesizing polypeptides. Option B, it carries amino acids to ribosomes. Option C, it is a constituent component of ribosomes. Option D, all of these. So, which is the right option here? That is, which of the following are the functions of RNA? Is D is the option here because it acts as a carrier of genetic information, that is messenger RNA, right? So that is a function of RNA, and it carries amino acids to ribosomes, that is an adapter, tRNA. Okay, so that is also a function of RNA. And option C, that is, it is a component of ribosomes. Is this rRNA is a main component of ribosomes. So all of these are the functions of RNA. So D is the right answer. Dash has the shape of a clover leaf. mRNA, tRNA, rRNA, and DNA. Is tRNA. B is the right answer here. And dash is a process in which information is carried from RNA to DNA, and it is happening in some viruses. That is RNA to DNA. So option A, transcription. Option B, replication. Option C, reverse transcription. And option D, translation. Yes, option C is the right answer because the process in which information is carried from RNA to DNA is called reverse transcription. Okay, so this is happening in some viruses. So C is the right answer. So I hope you are clear with this right till now. So we completed about the structure of DNA, then packaging, and how they prove that DNA as a genetic material, and also we discussed about the stability of DNA and RNA. So regarding this, only the problem-oriented questions are the percentage composition of the different nitrogenous species. So you focus on that, and other than that, everything will be direct questions only. So if you understand the concept, and you can write any type of question, you can answer. Okay. So tomorrow we'll start with the central dogma, that is about replication. Okay, so how the DNA is replicated. So form more copies of DNA. So what are the enzymes which are involved in the replication process? So you go through it, and if you have any doubts, maybe you can discuss. And tomorrow we'll continue the replication. So clear? So no doubts, right? So we'll continue tomorrow. And also you complete those questions and we'll discuss the answers for those questions also. That is principles of inheritance and variation. Okay. So we'll discuss that tomorrow. Okay then. Thank you.